Students, up till now we have talked about how microbes are present everywhere. So, they are ubiquitous in nature, or essentially ubiquitous, because wherever there are even slightly hospitable conditions, microbes will survive. Some microbes will thrive there. And the other thing we have learned is how microbes are integrated with our public health and our own well-being. Not only in terms of their pathogenicity, that some microbes are pathogenic, but also that some microbes, some commensal microbes are necessary for good health. For example, in the previous lecture we talked about if the gut microbiome is healthy and is adequate enough to promote lean body mass, then the person or the rat is likely to have a lean body mass. On the other hand, if this gut microbiome is affected and it changes to it changes its profile, then it is quite possible that now the new profile will not support a healthy lean body mass and will affect the body mass index of the person or the rat. We also learned about IBS, which is irritable bowel syndrome, and how it is now we are suspecting, and we have good reasons to believe it is largely governed by the microbial flora inside our gut. Obviously, we do not know we have not pinpointed the exact reasons which affect the constitution of microbial community in the gut in the first place. And if you remember, one of the examples that I gave you in the one of the previous lectures was when the cow or a ruminant is diet of the ruminant is shifted from a grass or hay based diet to a grain based diet, then that results into acidosis, because now the microbial community is overtaken by Streptococcus bovai, which utilizes the starch in the grains and causes acidosis, which can be fatal for the ruminant. Similarly, we are trying to find out what are the factors that affect our microbial community, also the microbial communities around us, how they interact with the microbial community within us, and how that affects our public health and our personal well-being and health. Now, with that we come to the concept of exposome that I will be talking about today. So, what is exposome? If you look at it, is spelled as EXPOSOMES, and it is pretty much similar sounding as other omic words such as metagenomes, so metagenomics, or proteomics, metabolomics. So, omics is whenever we talk about the entire if you are talking about metagenomics, for example, we are talking about all the metagenes that are present in the sample. So, omics means all encompassing. So, when we are talking about exposome, there are two words here, expose and the other is ohms. So, ohms means all encompassing, and expose comes from the word exposure. So, everything that we are exposed to, our environmental systems and public health systems are exposed to, comes under the exposome. So, let us take a look into exposome, and I must tell you that exposome, the conversation on exposome started in 2005, and by the time we started the conversation about exposomes, we already knew how our gut microflora affects the way our mind works, and the way our mind works affects our gut microbial flora. And we realized that apart from this particular condition also, the microbial community in our environment, we already knew that in our environment, in our bodies, and everywhere else affects everything else. So, we had already appreciated the importance of microbes, the importance of our exposure to them, and we had started questioning certain practices as indiscriminate utilization of antibiotics. So, if for example, if this intestinal wall is exposed to antimicrobials, then these microbes will die out, which will prevent the proper fermentation of the food as it is supposed to be in the hind gut, and that will impact the kind of messages that the gut sends to the brain and the way the brain feels. Also, when these microbes have been removed using antimicrobials, then the pathogens have a higher chance of surviving where there are no living microbes waiting to compete. So, let us look here in this particular diagram that I shared in the previous lecture. We talked about the interplay between digestive tract, which is our intestinal wall here represented by this, and our central nervous system, which includes our neurons and our brain. And we talked about how the signals produced by our gut are given to the brain used through this gut brain axis. So, the gut is communicating with brain through the vagus nerve, and the brain is regulating the digestive tract activity. Now, note this thing here, effect of environmental stress on the brain can have lasting impact on gut health. So, the environment might affect the brain. For example, it is too hot, too cold, or stressful, or lack of sunlight, or whatever, or the circadian rhythms are changing, or whatever other reasons are that we do not know yet, or maybe there are maybe the body is exposed to other contaminants. So, body is exposed to other contaminants which have stressed the entire body. All of these exposures, these environmental conditions, exposure to contaminants which may be chemical, biological or physical in nature, will impact the microbial community flourishing within our bodies, and that will impact our gut health in this schematic, and will also impact our well-being in other respects. And when that happens, now our for example, in case of gut health, we are more susceptible to diarrhea, and we are more susceptible to sickness in general. This can be because we do not have the good microbe, and it can also be that now pathogenic microbes have a higher chances of infiltrating these places and colonizing them. The next thing we also talked about in the previous lecture was how the behavior, these are the behavioral effects, how they are dependent upon our microbial community, and how our behavior also affects our microbial community. Now what we know is that it is not just our mental state of being, but it is also the environmental state of being. So, what is missing in this picture and has been added since 2005 is the environmental stressors. So, for example, now here I am exposing the body to endocrine disruptors, or if I am exposing the body to low grade endocrine disruptors, less amount, or if I am exposing bodies to disinfection byproducts. Now, in this case, the microbial community here, the functioning of brain, everything will get impacted. So, it is not just if I am being exposed to pathogens or not which decides my susceptibility to develop diarrhea, but also what else I am being exposed to in the environment, how stressed and vulnerable my system is. And this is what brings us to the exposome, which is the topic for today's lecture and the lecture after this. So, the exposome concept, as I mentioned, it came in 2005, and this was as a result of public health studies. Now, conventionally the public health studies, they look for cause and effect. I mean, there are many different kinds of studies we can do, but in many epidemiological studies we are trying to figure out the cause and effect. For example, in the famous London cholera case, we wanted to find out what was the source of cholera that spread the cholera outbreak in London, and then we found out it was hand pump, etc., etc. So, we want to find out the source. Now, by this time, many studies in public health had demonstrated that it is not always one single source that makes all the difference. At times the body, the health of the community members is already compromised, and then there is a trigger which allows the pathogen to infiltrate and to colonize the bodies and make them fall sick. So, when this happened, then when this understanding gathered momentum, then in 2005 in a seminal paper, the scientists started talking about the totality of human environmental exposure from conception onwards complementing the genome. Now, I should also mention that when we are talking about this aspect of public health, lot of research especially by, for example, Dr. Gillian Banfield in US and other similar researchers had shown that the early colonization, so when the infant is about to be born, the infant's body is not colonized by microbes, but when the infant is brought out in the open in the environment, depending on whether it is C-section or normal delivery, the child will be differentially colonized by microbes. Now, this colonization of microbes, and then so this is microbial exposure, and then we might have other things such as chemical exposure. For example, now we know that there are many flame retardants that break the blood milk barrier and come in the breast milk. So, now the infant might be exposed to these flame retardants, bromine based flame retardants, which are not good for its health and growth. So, all the exposures starting from not just birth, but even conception. For example, if the mother was exposed to smoke, alcohol or drugs or some other contaminant like flame retardants, then many of them will pass through the blood and blood barrier and blood fetal barrier and damage the fetus or affect it. So, right from the conception, what exposures were involved and how they have affected the genome. And the reason why they started talking about this is that before this 2005 seminal paper, there was it was believed by many microbiologists and environmental microbiologists that it is our genes that determine our ability. So, for example, if now people are talking about there should be a gene for alcoholism, a gene for homosexuality, a gene for this, a gene for that. So, let us say a putative gene for alcoholism, and what the research started gathering up by 2005 was it is not just the presence or absence of the gene, but a lot about regulation of the gene. There is a lot of things talking about how the environmental exposure complements the genome, and this is where the exposome concept came from. What it does is it calls for providing a description of lifelong from prenatal period exposure history. So, right from birth, even birth, from conception to this current moment, what are the different chemicals you have been exposed to, what are the different kinds of smoke you have been exposed to, environmental stresses. So, body accumulates the stress from all of that. So, there are two kinds of responses or reactions from the body when it is exposed to a xenobiotic or high amounts of any concentration or any contaminant. One is acute effect, the other is chronic effect. So, acute effect will happen on a short term range. For example, if I am exposed to mustard gas, high amount of it, and the acute effect would be fatality. However, if I am exposed to trace amount of mustard gas, very little, not enough to kill me, and maybe the exposure happens once or twice here and there, nothing happens, I am like, okay, I am cool. Nothing happened for 1 day, for 1 week, for 1 month, 1 year, 10 years, but then by the time the 10 years have passed, it is quite possible that that minute exposure to mustard gas 10 years ago started causing changes in my genes, which over time have had a cascading effect, and now I am developing diseases and cancer, for example cancer, that are now lethal to me. So, we can have both acute and chronic effect. As I mentioned earlier, the public health studies were often designed to be pro-acute conditions because they want to find out cause and effect. So, ideally we have a cause and it is causing an effect. So, we have a pathogen or we have an environmental problem. For example, a particular kind of fungus is growing in the sludge, so we are having a particular effect, right? Or protozoa is increasing in the sludge, so we are noticing these problems with our sludge. So, these cause and effect relationship investigations work really well if the time lag between cause and effect is not significant. So, time, if it is small, if the effect is acute on an acute scale, that is a small time scale, then these studies are possible. But many a times the cause is so remote in time, it has happened in such distant past that we are looking for the nearby causes. Cause 1, is this cause, is this the cause, is cause 3 the cause, and none of them are singular causes, but a cause that happened way past and past might be. And that is the reason why people are talking about since conception till until now. But as you can imagine, these studies are very difficult to do, and this is where our environmental microbiology comes into picture. Because for example, let us say I have a human being here, and this human being eats something which is not good for his or her health, or drinks water laden with hepatitis A and E and gets jaundice. There is another person who does not get jaundice. What determines the susceptibility of person to get jaundice and not get jaundice? Now, in this case, we want to look at the exposure to all microbes and exposure to all chemicals and all physical conditions and emotional conditions and social etc. conditions, all of them which come under exposome, that make one person more susceptible to hepatitis A or E infection and the other person not so susceptible. So, if we can figure out this, what causes a person to be susceptible to an allergy, to a chronic condition, or even to infections, then we can figure out novel ways of treating our patients and also taking care of our environment. So, let us look into it. In order to understand our exposome, we need to have information on 3 different domains. The first is your general external environment. For example, you need to have general information about external environment. For example, is this person from which developing country or developed country? Are these people from rural area or urban area? What kind of occupation they do? What kind of climate they live in? Do they live in hilly terrain? Do they live in hot tropical climate places? what is their social status, what is their economic status, what is their educational status. So, we need to know the big things. Now, we know that the mind affects the gut, mind affects the microbial community and well-being of the gut. So, we also know that social factors, emotional factors, education, etc., would affect the microbial community in the body. Next thing, we want to know very specific external environmental cues. For example, we want to know, was there any difference in their diet, in their physical activity, consumption of tobacco, or did they have any prior infection. For example, this particular person who is susceptible to getting hepatitis A or E after drinking contaminated water, that is developing jaundice-like symptoms after drinking contaminated water, then maybe this person already had certain amount of liver damage or had some certain amount of compromise in their immune system, and whereas the other person did not, and that is why this person had symptoms of liver damage and the other person did not. So, now we want to find out very specific external conditions such as prior infection, tobacco, physical activity, occupation. Now, the next thing we want to understand is, so we have figured out the external environment, the general and the specific. Next thing we want to understand is your internal environment. For example, what are the metabolic factors in your body? What are the different characteristics of your serum, your blood, your microbial communities, the juices in your body? What is your gut microbioflora? And what we are finding out that the gut microbioflora affects not only gut related diseases and allergies and chronic conditions, but also has impact on the entire health. So, we also want to see the degree of inflammation in the body. Apparently, when body suffers from high inflammation, it is more susceptible to certain diseases. Similarly, with oxidative stress and aging, we already know that elderly population are more susceptible to infectious diseases. So, maybe this person who is susceptible is basically elder. So, you want to understand, or maybe it's the same age as the other person, but the body has aged much more in this case than in this case because of some specific or general external environmental or internal environmental conditions. So, once we have figured out all these three, only then we can go and start identifying putative causes, and then we get more data, we can perhaps gather evidence in favor that okay, this cause seems very likely. So, we notice here that the exposome takes care of your external environment. So, this is your external environment. And this is your general external environment. So, it is very general, what kind of place you live in, what is its demography, its economy, social life, and what is your urban environment. And then we talk about specific external environment. So, what is the specific external environment of the person, and then we look at the internal environment of the person. We can do, right now we are doing 3 different analyses for internal environment. One is the transcriptomics, if you remember. So, omics means all, transcriptome comes from transcription. So, all RNAs in the body of the person or in the gut microbiome of the person are sequenced. So, we understand what genes of the body are being transcribed, and then we look at the proteomics. So, we are looking at post translation. So, we want to know all the protein structure that are present in the gut microbiome or in the body, and then we work at metabolomics, which looks at all metabolites, typically done through liquid chromatography and mass spectrophotometry. And nowadays we apply UPLC-MS or at times UPLC-MS for doing metabolomics. Now, this is your understanding of the internal environment. Using data from the general external environment, specific external environment, very specific internal environment, over the life course and under the dimension of life course, we understand better now the health risk and impact assessment. What is this person likely to get as a disease? Now, here I am talking about personal, but we can also isolate what are the specific external environmental cues or general external environmental cues or internal environmental cues which result in a particular disease, and what is the probability we will get a disease if we are using this consumer product versus another consumer product. So, this exposome studies are very useful for risk assessment and health impact assessment. Alrighty. Now, why we need them? Just a revision of that. We know this now that the genetic variants only explain a small fraction of the diseases that we are noticing, especially in chronic diseases, allergies and other diseases such as type 2 diabetes and obesity. We know that it is mostly your wide range of environmental and lifestyle risk factors that contribute to triggering these genetic factors. especially in case of even viral infections, allergens, diet, urbanization, all these affect the probability of getting asthma and other allergies. However, we have very limited knowledge on the environmental factors that contribute to allergic diseases. We know that there is a difference, but there is an effect, but we do not know which environmental factors have how much effect. And this is where, as applied environmental engineer, you come into the picture, or applied environmental microbiologist, however you want to call yourself. You come into this picture and you try to understand what are the environmental cues, environmental factors that affect our microbial community such that we are more susceptible to a particular disease, whether chronic or acute. And in doing this kind of study is actually, this is quite a hot research topic right now. Again, for students who are interested in research, please look up Exposome. In this week's homework, we have a lot of questions about Exposome, and I hope that will make it more clear when you are trying to answer the assignment in this week. Alrighty. And why do we not know about environmental factors? A, because this understanding is a recent development. The other thing is the main environmental risk factors are unknown still, because we do not have investigating tools to identify them, to understand them. And the other is that environmental exposure is time, it varies with time. We do not have history of the person. It is very hard for us to develop the history of the person now, to catch back the history of the person who is sick now or healthy now. What we can do is we can take babies, like what did Dr. Gillian Banfield did was, in one of her studies, she took she understood the microbial communities on skin and in the gut of children right after when they are born, and then noticed it over a period of time. So, how the microbes in our environment and in the mother's body colonized the infant and how that affected the health of the infant. So, these kind of studies are exposome studies, and the important thing is that they consider the lifelong exposure history, and therefore they want to notice how change in exposure over time will affect your health. I will give a very simple example. We live in India, and in India air pollution is one of the largest killers. It's some reports say it is the 4th largest killer in our country. Now, most of these reports are based on direct impact of air pollution. For example, if I am living in a very polluted city, for example Delhi, and especially during its highly polluted times, then it is likely that my asthma might get worse, I might develop lung irritation and finally lung cancer, and then I die. And then people say, okay, air pollution, one more person died. Now these are, it is very easy to observe them, but I live in Roorkee. So in Roorkee, when I am being exposed to not hazardous levels of air pollutants, but still considerable amount of air pollutants over the course of my life. Now, I might be more susceptible to certain other infections, for example, pneumonia or other viral bacterial infections that people exposed to the contaminants that I am being exposed to at a small concentration are susceptible to get. Now, this kind of now if I were to die in few years from those diseases, then we will not label air pollution as a reason, but we would call the immediate cause, which could be a certain kind of cancer or which could be a certain kind of bacterial or viral disease, as the cause of death, and thus the amount of deaths due to air pollution are underestimated. Now in this case, if we know the chronic exposure history of the patient, the person who is dead recently, and we know that what is unique about this person, okay, this person lived in a Roorkee town, highly populated town, and was exposed to this amount of these contaminants, which people, and then they can do a nice study, cohort study or case study, and figure out whether what contribution it was of the air pollution I was exposed to on a daily basis that resulted in particular disease. So this is where the exposome comes into picture. Now another, staying with the air pollution, another thing is many of our working class members now spend more time indoors than they spend outside. Most of the middle class families and also upper class families, when we talk about economic classes, in fact most of our countrymen now spend more time indoor than they would spend a few years earlier when we were largely rural economy. Now when we are spending time indoors, we are being exposed to volatile organic compounds and other contaminants that are found mostly inside the house and not outside. Now in this case, our bodies are exposed to different contaminants than the person who sits most of his time outdoors and are exposed to the contaminants in the air. So now I might develop some disease because I spend more time indoors that do not have a very good correlation with the contaminants found in the local Roorkee air. But if I look at my personal exposome, then you will know, oh, Gargi developed this disease because of XYZ exposure. This is again where the lifelong exposure history plays very important role. So let's read this. To date, most environmental health studies assess the environmental exposure at a single time point and therefore did not cover or may not have covered the highest risk exposure window. So now there are some really big research centers that are working on to gather data on exposome. One of them is the Hercules Exposome Research Center, and they are funded by NIEHS, National Institute of Environmental Health Sciences. And look at what they have written for the environmental exposome. The environment, broadly defined, plays a major role in health and disease, but has been underrepresented in research community. Dear students, this is again it's very important for us to understand applied environmental microbiology, right? And this is also the scope of this particular subject that we are studying now. The exposome provides a potential vehicle to better incorporate the environmental component into study of disease and health. Once we understand our environment better, once we understand the environmental exposure better, we will be able to explain why people are developing a particular disease and are having specific health challenges. Our long term goal is for Emory, the place where they have this center in US, Atlanta. Our goal is to play a leading role in discovery, evaluation and application of exposome, and now they have other kind of information about their center. But these first two lines are very important for you to understand. Next, there is this human exposome project which collects all data on human exposome and summarizes on their website, and then there is an Exposomics. They are doing some very good research in water pollution, air pollution, in biostatistics and in omics. So let's look at that. So when it comes to air pollution, they are finding out that we need to understand what is a person's personal level of exposure to different contaminants. And if you look at this particular figure, and this is from one of their studies where in their backpack they had a bespoke rack inside this backpack that held sensors and batteries. Now these sensors sense different kinds of contaminants. They sense, what were they? There was battery for supporting the sensor. There was a pump, a PM 2.5 pump based on cyclone movement. What it would do, it was collecting air and measuring real time amount of PM 2.5. There was a UFP disk mini which measured other kinds of contaminants, kept the information, stored it into SD card. So this information was available, it was shared with the researchers. Now there was also separate the candidates who were carrying this backpack. So people were supposed to carry this backpack wherever they went throughout the day. For example, I don't sit in this room for entire day or every day even, but I go to different departments, different places. I also take a walk around the campus, I go to market. So as a typical person, you also have a lifestyle and you don't stay confined within a room. So wherever you go, you are being exposed to the contaminants present in that particular environment. Now that's the importance of why we use backpack for storing our sensor and collecting real time data. Now the person is also given a GPS and accelerometer for locating the person. Okay, when person was in market, the person was exposed to these contaminants. When person was in this room, this contaminant. Accelerometer also helps us understand the velocity and thus validate the data that we are getting from GPS. Now this data is again shared to the software, the bespoke software. Together this data is sent to the process data. Now when they did this study, they found out that the exposure for different kinds of lifestyle is distinct, and also when the exposure changes, the the effect of contaminant on health also changes. Now this is based on personal monitoring. Now what they did was they collected data based on personal monitoring over time, how different kinds of people had different kinds of exposure, and now they also have satellite data. So this is satellite data usually will capture your outdoor data. So what they did was in this study, Exposomics people, they tried linking that, okay, if we have this amount of PM 2.5, here PM 2.5 is very low in the Scandinavian countries, north of Ireland, Scotland and England. Now in these countries where in these regions where PM 2.5 is very low, let's find out what the personal exposure is. So satellite data is easier to get, personal exposure studies are hard to do, more expensive, more labor and capital intensive. So, if I know, for example, this region has higher exposure to PM 2.5, what about people who are caring about their lives here? So, basically what I am trying to understand that how does the ambient air quality affect the exposome, not only when the person is spending time outdoor, but also when the person is spending time indoor, because eventually the air will find a way to seep in. Now, this is a very nice, interesting ongoing study, and I encourage you to follow Exposomics and understand about the latest research that they are doing. Now, what they look here, these are the air pollution measurements at home when the outdoor monitoring was done. So, the outdoor monitoring is given to you by satellite, and the backpack indoors is basically the person where the person is sleeping. The person will put backpack right there, the person will move around the house, maybe go out, catch a bus again indoor, and then go to office and come back. So, when they compared this in the same location, I have some information from the satellite. I also have information on when I am doing personal exposure monitoring. I know the space-time pattern of exposure. For example, person is journeying, person is at home, at work, and when I do this, what I am getting is they developed a very nice model, land use regression model for exposomics, which map the concentration of outdoor air pollution. Now, currently they are trying to link all of this, and hopefully we will get very meaningful data on how the exposome varies when ambient air quality varies, and how that would affect the public health. So, dear students, this is all for today. In the next lecture, we will go ahead and talk more about exposomes and how they affect our health and what are the latest frontiers in research. Thank you very much.